A warm welcome to you all. Welcome to our 22nd INFOGEST Viva webinar, where we seek to promote your PhD work and disseminate it further. Today with us is Dorine Duijsens, and Dorine has obtained her PhD in Bioscience Engineering last year in August with a dissertation entitled Steering Digestion of Lentil-based Foods Through Structure Engineering and Advancing In Vitro Digestion Methodologies. She is currently working as a postdoctoral researcher in the Laboratory of Food Technology at KU Leuven in Belgium, and she's working on food and meal design for specific groups adapted using adapted in vitro digestion models. So today we will listen to a 20-minute audio from Dorine, which is pre-recorded. And thereafter, we are going to have a live Q&A session. So please do pop your questions in the Q&A box down below, and Dorine will be happy to answer those. So now we will switch over to the pre-recorded video from Dorine. Thank you.
Good afternoon. My name is Dorine Duijsens, and I recently obtained my PhD in Bioscience Engineering with the steering digestion of lentil-based foods through structure engineering and advancing in vitro digestion methodologies. Now, since today is a 20-minute presentation, I will focus on the first part, so steering digestion of lentil-based foods through structure engineering. Let us dive in. Why did I work with lentil-based foods, and why did I want to steer their digestion properties? Well, let's first have a look at many of the food recommendations. This is an example of the Flemish government, and they're asking us to incorporate pulses into our diet at least once a week, or replace meats by plant-based protein sources in around half of our meals. That's partially due to their interesting composition. Pulses are rich in protein, dietary fiber, and slowly digestible starch, and I will talk more about that starch later. Pulses are also recommended in the context of sustainable diets. This is an infographic by the European Food Information Council, but pulses are also incorporated in the planetary health diet, where they recommend us to eat 75 to 100 grams of pulses per day. So that sustainability can be attributed to a lower water usage for production as compared to other protein-rich foods, and the fact that pulses can fixate nitrogen in the soil, improving the soil quality. Greenhouse gas emission for 100 grams of protein is much lower for peas and other pulses as compared to many other protein-rich foods. Pulses have another very specific property that until a few years ago was mostly studied in beans. If we look inside the seed halves or cotyledons, we will find that the tissue is built up of cotyledon cells, which are linked together via a mid-lamella, which is rich in pectin. Inside the cells, we find the most important macronutrients, starch and protein. Now, when beans are cooked, the pectin in that mid-lamella, gluing cells together, is solubilized and the linkages between the cells weaken. When these cooked beans are then chewed or mixed or disintegrated by any other means, the cells separate, yielding individual cotyledon cells. Why do we care about these cells? Well, these cells are made up of starch granules entrapped within a protein matrix, in turn entrapped by a cell wall. And both that cell wall and the protein matrix can act as physical barriers, slowing down the diffusion of digestive enzymes through the cell wall and the protein matrix to the substrate, which in this case could be the starch granules. That leads to a slower starch digestion, and that has been linked to several consequences. For example, it has been linked to a slower increase of blood glucose upon pulse consumption, responsible for the medium glycemic index of pulses. But the slower nutrient digestion has also been linked to satiety, and the slower starch digestion could lead to more resistant starch reaching the colon, where it can be fermented by microorganisms. While the recommended consumption of pulses is quite high, this is not at all reached in many countries. And that can be attributed to the typically beany flavor of pulses that a lot of people do not enjoy, or their very long preparation times, which are not convenient. Now, in my research, we wanted to find ways to increase and stimulate pulse consumption, and for this, I selected lentils to work with, because these have a less typically beany aroma, but also because they require shorter cooking times to be palatable, which is more convenient. So on the one hand, we could try to stimulate lentil consumption by, for example, incorporating them in recipes of stews, salads, soups, etc. However, the approach that I took in my PhD is to try to incorporate pulses, and in this case lentils, into the formulations of everyday foods such as pasta or crackers. So, all of that introduction to explain to you what I mean by structure engineering of lentil-based foods to steer digestion properties. So we have established that if we cook lentil seeds and then disintegrate them, the most prominent microstructural fraction is that of individual cotyledon cells, and these have interesting slow digestion properties. However, if we look at most pulse-based food products you can find on the market today, they are mostly made of raw milled lentil flour. And when these raw pulse seeds are disintegrated as such, the cells do not slide off of each other, but they break open, releasing all cell contents. And as you can see, the starch and protein released from the tissue in this case, they are completely free and therefore expected to be more readily accessible to digestive enzymes as a consequence. So based on this insight, we questioned ourselves if we could use targeted processing with specific processing sequences to obtain lentil-based foods with specific microstructures that had also linked typical macronutrient digestion patterns. And to answer this question, we needed to investigate how processing affects the digestion of lentils, and in more detail, how processing affected the microstructure of lentils and how it in turn affects the digestibility thereof. So to investigate that, we selected 3 cooking times at which lentils showed a palatable texture. Upon cooking, we then mechanically disintegrated the seeds to simulate domestication in a standardized and reproducible manner. Since we expected individual cotyledon cells to be an important microstructural fraction of these cooked seeds, we also investigated the cells as such, and we did that upon a wet sieving step where we isolated the cotyledon cells. In that way, we ended up with 6 different samples, of which we wanted to investigate the microstructure, but also the starch digestion kinetics. The microstructure was analyzed using microscopy and laser diffraction to look at the particle size distribution. On the other hand, we also simulated the digestion of these 6 samples using the static INFOGEST protocol. We wanted to investigate the time-dependent digestion of starch, or the kinetics of starch digestion, and to do so, we started the digestion reactions in a set of tubes, mimicking subsequently the oral, gastric, and small intestinal phases. We then incubated that set of tubes at 37 degrees under a constant rotation, and at predetermined time points, distinct test tubes were taken out of the incubator one by one, after which the enzymatic reactions were stopped. The digests that we obtained for each of these time points were used to study the amount of starch and also protein that was digested and the amount of metabolites that were released in the supernatant due to enzymatic activity. And that was used to calculate a percentage of digestion. Now, how does such a digestion tube typically look like? Well, we will get a supernatant and a pellet fraction, and we consider the solubilized metabolites to be potentially bioaccessible. So for the case of starch digestion, we quantified the reducing sugars formed during digestion based on a colorimetric interaction with the DNS reagent, giving an absorbance as 540 nanometers, and in that way a darker color coincided with more reducing sugars formed upon digestion and a higher level of starch digestion. Let's have a look at some of the results we obtained for cooked lentils and cotyledon cells as a function of cooking time. First, this is the particle size distribution of the individual cotyledon cells, and this was identical regardless of the cooking time. Under the microscope, it looked a bit like this, so individual cells with a seemingly intact structure of around 100 micrometers in diameter. For the whole disintegrated cooked lentils, we found a different profile, also with a clear peak at 100 micrometers, indicating the characteristic microstructural fraction of individual cells, as confirmed by microscopy also. However, we also found some other fractions. This can be explained by the presence of larger particles, such as seed coat particles, but also some clusters of cells that were still sticking together, and the presence of smaller material, which indicated the presence of some broken cellular material with some free starch and free protein. Now, what we can see is that with increasing cooking time, the fraction of larger particles, and we expect this to be clusters of cells, decreased in favor of the individual cotyledon cells. So these clusters actually disintegrated into individual cells as a function of cooking time. So then we investigated the impact of these microstructural properties on the starch digestion kinetics. For the individual cotyledon cells, with all a very similar microstructure, we also found very similar starch digestion kinetics. That indicates that the starch digestion properties of these cells did not really depend on the applied cooking time. We could also visualize the gradual emptying of cotyledon cells upon Lugol staining of the starch. As you can see, at the beginning of small intestinal digestion, cells are colored dark blue, indicating they are completely filled with starch. With increasing digestion time, we still see some cells that contain a lot of starch, while other cells show a gradual emptying pattern from the outside in, with the colors evolving from dark blue towards brown and lighter yellow. At the end of the digestion simulation, we even see some cells that seem completely emptied, while other cells still show some dark blue color, so a significant amount of starch that is present. If we then look at the starch digestion in the cooked disintegrated seeds, we see that the rate of starch digestion, but also the final level that is attained, increases with cooking time. That can be explained based on the microstructure, with more clusters present in lentils with a shorter cooking time. And these clusters, we hypothesize they can slow down digestion even more as compared to individual cells due to the presence of multiple digestion barriers. With increasing cooking time then, these clusters more and more separate into individual cells. We also observed that the behavior we see for longer cooking times for the whole disintegrated lentils overlaps with the behavior of the isolated cotyledon cells. This indicates again that this cotyledon cell fraction is indeed the most prominent microstructural fraction of the cooked lentil seeds at these cooking times, and that this fraction is also completely determining the digestive behavior of the cooked seeds. So, for now we found that if we cook, followed by mechanical disintegration, cellular intactness is maintained, and that cellular intactness leads to a slower starch digestion. Now the next question was, can we reverse engineer that process to obtain a food, lentil food, with targeted digestive function? To do so, we wanted to make lentil-based food ingredients. We did this by pre-soaking and cooking lentils for a short cooking time, 15 minutes, and then on the one hand, we isolated the cotyledon cells again through wet sieving and freeze-dried that into a powder. That was a flour consisting of cotyledon cells with encapsulated nutrients. On the other hand, we also generated a food ingredient from the whole pre-cooked lentils without generating an additional waste stream upon sieving. As a reference, we generated a flour by raw milling whole lentils, hypothesizing that the microstructural organization of the seed would get completely lost. Let's have a look at these flours and more specifically at the microstructure thereof. If we look at the isolated cotyledon cell powder, we see again one peak around 100 micrometers indicating the presence of individual cotyledon cells. For the whole pre-cooked powder, we also see a peak around 100 micrometers and the micrographs also show us individual cotyledon cells. However, we also find larger material indicating the presence of cell clusters or seed coat material and some smaller material which is not so well visible on this graph, but indicating that some starch granules and protein bodies might be released upon cell breakage. The raw milled flour then showed a different microstructure, with a peak at smaller particle sizes indicating the presence of free starch granules and protein bodies and cell breakage, and the peak at higher particle sizes indicating the presence of seed coat fragments, but also some tissue fragments that were not completely disrupted upon milling. The digestion properties of these flours were investigated in a model food system. That was a 10% dispersion of the flour in water. Digestion was then simulated as we explained before, and starch digestion was assessed spectrophotometrically again in each of the obtained digests as a function of time. Let's first have a look at the digestion of the starch within the raw milled flour. We see one first very rapid phase of starch digestion reaching around 80% that can be attributed to the free starch granules present in the raw milled flour. A second slower phase can be attributed to the presence of some tissue fragments that were not completely disrupted, delivering an amount of starch that is a bit more slowly hydrolyzed. The other two flours showed clearly attenuated starch digestion, with the isolated cotyledon cell flour showing just one phase of very slow starch digestion that is caused by the presence of intact cotyledon cells with the cell wall and the protein matrix as barriers for starch digestion. The whole pre-cooked lentil flour then showed biphasic starch digestion, which was also much slowed as compared to raw milled flour, and that indicates the presence of multiple populations of starch with a different susceptibility to digestive enzymes. In this case, that can be understood by the fact that some free starch granules are present. Starch is also present in individual cotyledon cells, but also in clusters of individual cells. So to recap, by choosing our processing sequencing in a smart way, we could get lentil powders with very distinct microstructures, so with and without cellular intactness. And we could see that that also led to distinct digestion properties. So this was only done in a model system, and of course we wanted to test if that concept also works in a real food system. And in this case we wanted to make a lentil-based pasta with intact cells. We decided to make pastas using the 3 distinct lentil flours that I just showed you. However, to make a continuous phase that is necessary to make this macrostructural pasta, we needed some free starch and protein in our flour blends. That's why we could not work with pure isolated cotyledon cell flour, but we had to work with blends. We worked with 100% raw milled flour as reference, and then with blends where 30% of the raw milled flour were replaced by either one of these two ingredients. For the choice of this substitution degree, I want to refer to a recent paper of my research unit where we investigated the impact of the dosage of cellular bean flour on digestion properties. But back to our lentil-based pastas. Of all the flour blends, the pasta were made by adding moisture, kneading the dough, shaping the pasta and then air drying it, followed by cooking of the pasta to their optimal cooking time. Now, of course, we wanted to know if that cellular structure was maintained upon all of these steps of the pasta making and the cooking and even the digestion afterwards. So we investigated this microscopically. For the 100% raw milled pasta, we could observe a continuous phase made up of starch and protein and some seed coat fragments that were just dispersed in that continuous phase. For the 30% isolated cotyledon cell flour pasta, we found the same, so the continuous phase and a few seed coat particles, but we could also find individual cotyledon cell in the structure. The same was true for the 30% pre-cooked lentil flour with individual cotyledon cells, but also some seed coat particles, and of course a continuous phase. So that was good news because we saw that the cellular intactness was maintained upon generation of the pasta and cooking of the pasta. Of course, the next step was to investigate the in vitro starch digestion kinetics of these pastas containing the innovative ingredients. To do so, we first cooked the pasta and cut it into small pieces to simulate mastication, and then digestion was simulated exactly as I've explained to you before. And in each of the digests that we obtained as a function of digestion time, we determined the amount of digested starch using the colorimetric approach that I explained previously. As you can see for this graph, starch digestion was significantly impacted by the incorporation of flours with cellular intactness. We clearly slowed down digestion, but also yielded around 20 to 30% less starch digestion by the end of the simulation. So again, due to the cellular intactness which prevailed even upon pasta making and cooking, the kinetics were significantly slowed down. This brings me to the conclusion of this work. We investigated that upon cooking of lentils, but also processing into cellular ingredients and secondary processing into a pasta, that the cellular integrity prevailed. And that cellular integrity affected starch digestion in each of the cases. So, if we want to conclude something from this presentation, it is that we proved the concept that structure engineering can be used to steer the macronutrient digestion, in this case the starch digestion, in lentil-based foods indeed. That's I conclude my presentation. I want to thank you for listening to me, and of course, I'm looking forward to hearing any questions you may have for me.
Thank you, Dorine. That was a really good presentation. Thank you very much. So maybe I will start with the first question, which is about the preparation of your lentil flour. Can you explain a little bit why is it that when you incorporate the cooking step before the mechanical disintegration, the cells do not break open?
Yes, well, that's a typical property of the pulses because their cell walls are so rich in pectin. So what happens actually during the cooking of the seeds is that the pectin solubilizes, and the pectin is very high in the mid-lamella. So if you imagine the cells, the mid-lamella is actually the part where they are glued together. So that pectin is very important in keeping that structure intact. But upon cooking, that is a polymer that's being solubilized, and that causes the weakening of the middle lamella. And as a result, if you then apply a mechanical force, this will be the weakest point of the tissue. And so when the tissue ruptures, it will happen at that location, and that will cause the cells to slide off of each other. You can compare it a bit with biting in a mealy apple and getting this granular feeling when the apple is too old, compared to a fresh one where you bite through it and the cells open and the juice comes out. And yeah, then the consequences, of course, for digestion are the fact that these nutrients are maintained within the cell.
Mm-hmm. And then the permeability of the cell wall. How is amylase able to penetrate? Are amylase able to penetrate through those cell walls, or do they have to break open for the starch to be accessible?
No, that's what I wanted also to show with the microscopy pictures. So that was quite remarkable, I think, to us when we first saw this. But these cells actually remain intact throughout gastrointestinal simulation. I've even done some fermentations of these pre-digested pellets. So I even tried to simulate the next step. And we see that the cells actually, for a big part, they remain intact. But if we then look at these Lugol stained micrographs, we really see the starch going away, let's say, or being reduced, the starch content being reduced from the outside in. So what we really expect that happens is that the amylases move inside the cell, break down the starch, and then the metabolites have to go out again before they can be absorbed. Of course, the mechanism or how this exactly happens is still a bit of a question, and it's also something that we are trying to answer now in following research. But indeed, we really think that the enzymes move inside before they break down the substrate.
Yeah, the microscopy images, they looked really, really nice, with the empty cells and we have a question in the chat here. I don't know if you want to add any more details to this. You have already explained a little bit. So it's could you explain how do you separate the cotyledon cells?
During the wet sieving, because I talked about the wet sieving a bit, it's actually just a stack of sieves that we use in the lab. And when we have cooked lentils and we mix them, typically done with a laboratory mixer, we have this kind of slurry, right? And that contains seed coat fragments, some clusters of cells, individual cells, and some broken materials. So we have materials of different size ranges in that slurry. So in our stack of sieves, we actually start with sieves with a large mesh size, and it becomes gradually smaller as you go down. So we load that sample on the top of the sieves of the sieve system, and then actually that whole tower is being shaken, and then and at the same time there's water flowing over the system. So as a result of that shaking and the water flowing through, the particles are separated based on their size, because as soon as a particle is too big for that sieve, it will remain on top of it. And just using the appropriate sieves and with some preliminary experiments to determine where the cotyledon cells can be found, where the clusters can be found, and what is too small to be cotyledon cell material, we just determined a range where we were sure that what was in there was just individual cotyledon cells. And for us, that was between 40 and 125 micrometers. And then, you just confirm that you actually have the fraction you want with microscopy or laser diffraction. Or
you also mentioned that you incorporated your flour in other products, in crackers.
no, it was just a suggestion. Now I know that. So for the proof of concept, let's say that I did in my PhD was a pasta, but of course that was actually just a proof of concept because it was also made with a manual pasta machine. I know that there's some recent work that's come out from another research group where they even were able to make a pasta upon extrusion. But you can, it could be used in many applications. I think cookies have been done, also breads have been done in the meantime.
What products do you think would be the most suitable? So it seems to work very well in pasta. Bread, for example, is another high GI food where there could be an interest, or muffins, or
the way I see it is it would be interesting to incorporate it in some high GI foods that we are consuming every day, because you could replace part of the highly digestible, rapidly digestible starch by a fraction that's less digestible, or more slowly digestible, I should say. So, I would be thinking of maybe wraps, breads, pasta could be a good option. More of the staple foods. I don't really see, I see the point of maybe adding it in a muffin, but that's not something that you are going to consume. Actually, it should be like a treat or something, not something you consume to get your like your steady energy supply, let's say. And there's already a lot of sugar in there, so I don't know if we replace 30% of the starch in there by a more slowly digestible source, if your blood sugar response will really be different because of the enormous amount of sugar that's in a cake or a muffin. So for me, it's more interesting for, like, for example, a bread or a muffin, like staple foods that you consume every day.
That's a good point. And have you done or seen work on different lentil varieties, for example, or because you used green lentils, right? Have you done any work with other types of lentils, or do you think there could be differences?
That's a very good question. I'm trying to work that out right now, actually.
Okay.
So,
we will be looking forward to those results.
I for the moment I have some preliminary results where I compared another type of green lentils with my green lentils, because the ones that I use are a Dupuis type, so it's a very specific cultivar that are quite small and dark green. So I compare them to the typical green lentils that are a bit bigger and lighter in color. And if we look at the digestion behavior for the starch and the protein, at least, of the cotyledon cells, they were quite similar. But I should maybe say that we, in that data set, also compared two chickpea batches that were both from the same cultivar from a different harvest year, and that showed quite big differences in the digestibility. So just to say that for everything I've seen on lentil up until now, looks kind of similar. But then again, for different pulse types, we have seen very big variability. I think there is still a lot of biological variability in the range of the pulse seeds. They have not been so much optimized as much of our cereals, I think. So a lot of biological variability is still present. I would not dare to say that what I found is maybe valid for all lentils out there. But the lentils that I have studied until now, they seem to have kind of a similar behavior.
That's very good, very interesting.
It's a cautious answer, but
I will be looking forward to know more about the other varieties. We have one last question in the chat. So it is about your digestion experiments. And the question, whether there is a specific reason for you to use 3 hours instead of 2 hours, as recommended by the INFOGEST protocol during intestinal phases.
Yes, of course. When I was in the beginning of my PhD, I started doing some digestion experiments, and so we really look at the kinetics of the experiment, right? So the approach that we take is for every digestion time point that we want to analyze, we use an independent digestion tube. And we stop the reaction at these specific predetermined time points every time in a different tube, so that all of these tubes represent the kinetics. Now, what I saw in the beginning of my PhD was that I was not yet reaching a plateau of digestion within that time frame of 2 hours, 120 minutes. And because I wanted to apply a modeling approach to be able to better characterize the time-dependent behavior and maybe estimate what the digestibility would be at specific time points, but also to compare the curve of the one to the other. Yeah, the fact that the plateau was not reached yet made it a bit more difficult to estimate our kinetic models, let's say. So that's why I just prolonged the simulation by 2 hours. I understand that these 3 hours is not what is recommended by INFOGEST, but just so that we would be able to make more accurate parameter estimates, let's say.
And maybe that also reflects a bit more what would happen, for example, if you had a more extensive, if you had an oral, like a more, I don't know, an oral phase in a semi-dynamic, maybe gastric phase, maybe the total extent of digestion would also be closer to what you have with the 3 hour intestinal digestion. It's difficult to predict, of course.
Difficult to predict. Yeah, I think with these starch-rich foods, if we really want to estimate the level of digestion that would occur in vivo, for example, I would go towards a more complex digestion protocol with an oral phase and saliva, and then a more realistic pH profile in the stomach, so that the saliva can also act for, let's say, a more realistic amount of time. Rather than just prolonging the simulation for that. I understand your point, but maybe if we really want to predict, we should go towards maybe more realistic digestion models. I think the models I use in this work are really very useful. The static models are really useful to compare the one food sample to the other, and really to try to understand why is this starch inherently more or less susceptible to enzymatic degradation than another one, more than really interpret the specific level, let's say. That's what I would say.
Yeah. I agree with you. Yeah,
That's nice.
I don't think we have any more questions, and we're also a little bit over there. So thanks very much, Dorine. That was a really good presentation.
Thank you for inviting me. It was really, really a pleasure to do so.
Thanks for your presentation. So next month, we will be back on the 7th of May with Sarah Perez-Villa, who did her PhD here in Teagasc and also in University College Cork. So that will be on the 7th of May at the same time as usual. So we would like to, before I finish, I would like to thank the INFOGEST Research Network and INRAE and Teagasc for supporting this initiative. And on behalf of my colleagues, Shannon and Cynthia and Ever, we want to thank everyone for attending, and we'll see you again next month. Thank you very much.